Hello everyone, this is Hariharan. In this video, we are going to see about maturation of B cells and T cells. First, we see maturation of B cells. The development of plasma cell and memory B cells are divided into three broad stages. First, generation of mature cells and incompetent of B cells, and the second is activation of mature B cells, and the third is differentiation of the activated B cells into plasma cells and memory B cells. These three stages can be divided into two phases, and the first stage is antigen independent phase and second phase is antigen dependent phase. Maturation. The generation of B cells first occurs in embryo and continuously throughout the life. Before birth, the yolk sac, fetal liver, and fetal bone marrow are the major site of the B cells maturation. After birth, the generation of mature B cells occurs in the bone marrow from the HSC. HSC is nothing else hematopoietic stem cells. The HSC first divide to form lymphoid progenitor cells, which then differentiated into the progenitor B cells. It's also known as a pro B cells that express a transmembrane tyrosine phosphate called CD45R. Pro B cells also express CD19, CD43, and CD24. A C kit are present on the surface of pro B cells. The pro B cells proliferate within bone marrow, filling extramuscular spaces between the large cisternites in the shaft of a bone. Cells requires microenvironment provided by the bone marrow is known as stromal cells. The pre-B cells express many of same marker that were present on pro-B cells. However, they causes to express c-KIT and CD43 and begin to express CD25. The IL-7 secreted by the stromal cells drives the maturation process eventually, including down the regulation of adhesion molecules on pre-B cells. So the proliferating cell can detach from these stromal cells. At this stage, pre-B cells no longer requires for the direct contact with stromal cells, but continues to require IL-7 for growth and maturation. Next we see the maturation of T cells. The migration of progenitor T cells from the early sites of hematopoiesis to the thymus take place at the about day 11 of gestation in the mice and in 8th or ninth week of the gestation is in humans. T cell maturation involves the rearrangement of germline TCR genes and the expression of various membrane markers. In thymus, the developing T cells are termed as thymocytes. These thymocytes proliferates and differentiates along developmental pathways that produces functional distinct subpopulation of mature T cells. When T cells precursors arise at the thymus, they don't express the signature surface markers of T cells as the T cells receptor CD4 and CD8. In fact, these progenitor cells have not yet rearranged their TCR genes and do not express the protein such as RAG1 and RAG2, which is required for the rearrangement. After arriving in thymus, T cells precursors enter the cortex and slowly proliferate. As these cells are c-KIT receptor for stem cell growth factor, CD44 and adhesion molecules, CD25, it is the alpha chain of the IL-2 receptor. These cells are called as DN2 cells. T cell development is a costly process for the host. These single positive cells undergo additional negative selection and migrate from the cortex to medulla, where they pass from the thymus into the circulatory system. After the gene rearrangement, T cell undergoes the thymic selection. Thank you.